Okay, so welcome everybody to the keynote session. It's my very great pleasure to welcome Dr. Drew Berry and Dr. Anders and Professor Anders Ynnerman, who are going to give the keynote jointly. Dr. Drew Berry is from the WEHI, Australia, and Anders Ynnerman is from Linköping University. Drew is a biologist animator renowned for his visually stunning and scientifically accurate animations of molecular and cellular processes. Trained as a cell biologist and a microscopist, Drew brings scientific rigor to each project, ensuring current research data are represented. Since 1995, Drew has been a biomedical animator at the Walter and Eliza Hall Institute of Medical Research. His work has been exhibited at international venues including the Guggenheim Museum, MoMA, and the Royal Institute of Great Britain. In 2011, he collaborated with the musician Björk for her album Biophilia. His many awards include an Emmy, a BAFTA, and the MacArthur Genius Fellowship. You can see his animations at WEHI.tv. And Professor Anders Ynnerman holds the chair in scientific visualization at Linköping University and is the director of the Norrköping Visualization Center. His research interest lies in the area of visualization of large-scale and complex data with applications in a wide range of areas, including medical visualization, space and astronomical research, as well as visualization in science communication. Anders is a member of the Swedish Royal Academy of Engineering Sciences and the Royal Swedish Academy of Sciences. In 2007, Anders was awarded the AXO Nobel Science Award, and in 2010, he received the Swedish Knowledge Award for dissemination of scientific knowledge to the public. In 2017, he was honored with the King's Medal for his contributions to science, and in 2018, he received the IEEE VGTC Technical Achievements Award. So please welcome these two illustrious guests to the stage for a fantastic talk that I'm very much looking forward to.
Thank you so much for inviting me and Drew to come here and talk about one of the most exciting projects that I've been involved in over the past years. And it's such an honor to be able to hear. What Drew and I will do is that we will take you on a journey. We will take you on a journey through scientific visualization, immersive experiences, but go all the way down to the micro level and look at some amazing foundational processes that are shared by every single living entity on this planet. And this is what's hopefully is going to make you feel a little bit of a wow feeling that's enough to make you want to come and look at the full show during the banquet on Wednesday. So we're super grateful for the opportunity to be here and share with you some of these things that we have been working on over the past 5 years now. I will start and give you a little bit of an introduction to the reason why we're doing this, and then Drew will take you on a journey through his amazing material that we have brought to the Dome Theatre. Now, the starting point for this endeavor is a pretty serious one. It's the problem that we have in the world with the decreasing interest among children and adolescents for science, technology, engineering, and mathematics. And it's shared worldwide, more so in some countries than other countries, and Sweden, where I come from, is certainly one of those cases where this is a huge challenge for society and for companies and for academia, for that sake, for the future, for the next generation. So there's a lot of support for us to be able to try to address this. And this is the reason why we have built the center where I'm based back in Sweden, the Norrköping Visualization Center, that has more than 200 people working there. And in visualization, a large research group, but also connecting with society in general. And the amazing fact that we actually, in this environment, we have our own museum, our own science center. And in the Science Center, we're trying to shortcut the distance between basic research in visualization and in applications and bring it out to the general public. And you know, for me, as a university professor, I'm so happy to be able to say that I have about 200,000 visitors per year coming to my research lab, and most of them are children. Next year, I will have 2 million, and I'm going to tell you the story why we're going to reach 2 million. Now, the academic component of this, you know, this mission that we set out to try to communicate science using visualization, we felt the need to try to frame it in some sense. And we have written a few papers on this. This sort of the migration, the shortcut that we are creating with visual representations from the foundational research all the way out to the general public and to the children, using the same methods, the same software, the same data. And I'm really sorry, but I invented this word called explanation. It's a terrible word, but it's the best word that I have to explain what we are doing, bringing exploratory visualization into the general public and using explanatory methods to do that, but shortcutting that distance. And there's a paper written on this if you want to read more about the considerations and the academic analysis of this transfer or convergence of paradigms that we're seeing in public spaces, in science centers, and in museums. Now, the main part of our center that we're going to talk about today is, of course, our Dome Theater. And it's an amazing facility. It's the first 6P laser dome theater in the world with amazing 3D capabilities and color contrast beyond what you could ever imagine. And we're very proud of that environment. And of course, I wish that we could all be there. But we have the chance to look at it in a really good planetarium dome theater tomorrow during the banquet as well. Now, what are we doing in these environments? Well, you know, we are working with trying to create these immersive experiences for our audience that's coming there. And it's immersive experiences that are explaining complex things, that are enabling us to explore the unknown, to engage the audience. The children that are coming, they're very engaged. You should see them picking stars and planets when they come into the Dome Theater. But it's also very entertaining. Now, what kind of evidence do I have that this is really happening? Well, and this is perhaps a little bit of a call to action for this community, is that there's a lot of people looking at various aspects of education and visualization, but that's very few studies that have been made looking at these big immersive dome theater planetariums and what kind of effect they have on an audience. But there is some evidence, and it's been a paper that came out in 2017 that showed the guidance that you can have, the scaffolding of knowledge that can happen inside of an engaging experience, and the learning outcomes that we are studying with this. But there's more. There's room for much more to do. And this community is well positioned to work on that. So let's have a discussion about this now. If you believe in this notion of immersion being a way to address the STEM or the STEAM issue among children and adolescents and bring science to the general public, and if you're excited about that, you go to a funding agency. So I did. I went to the Wallenberg Foundation, and we started up a project called Wisdom, and it stands for the Wallenberg Immersive Science Communication Domes. So in our small country, 10 million people, we have the Visualization Center, but we joined forces with some of the big museums and science centers in the country. And we were commissioned with the idea of building dome theaters and immersive experiences in these other places as well. So this is the starting point for this project. So apart from our own dome theater that we have in Linköping, we are now constructing dome theaters, or we're almost finished constructing dome theaters in 4 other places. One in the south of Sweden, opened up earlier this year. One in the north, opened up last year. One in Gothenburg, a gigantic one, and one in Stockholm that's going to open up in December. So now we have 4 new dome theaters in our own, and we're going to reach about 2 million visitors per year in these environments. And just to give you a flavor for the size of these things, here's the Gothenburg Dome Theater right there. It's a huge construction next to the amusement park that we're putting in. So we're bringing science, we're bringing our methods, our visualization technologies being brought into the public space in the context almost of an amusement park and the science center that's connected to that. That's the largest science center in the Nordic region. Now, we need to produce content for these dome theaters, and this is what we're going to talk about today, and what Drew is going to show you some of the aspects that he's been working on as well. But to do that, you have to reflect over what it is that you need to work on, and we have this sort of the notion, or what we call the success criteria, and I'd like this list of things. If you can put a tick on this list and say, I'm doing this, I'm doing that, I'm doing this, I'm going all the way through this, then you have a pretty good science story that you can tell. You have to be simple, it has to be unexpected to get some adrenaline in the audience, it has to be concrete, credible, emotional, it has to contain the science, and it has to be based on storytelling. And if you can tick out that list, you're probably in pretty good shape to start producing things. There are, however, some challenges. And you realize immediately that your competition is stiff if you look at the younger audience. You look at the children, right? And they are basically grown up on Hollywood productions and super nice animations from places like Pixar and the others out there, right? The budget for one of these things is astronomical. And if you produce a dome show, your budget is pretty limited. And this is a challenge, right? How can you meet the expectation of that very demanding young audience when they come to your facility? There you have to be clever, okay? This is a challenge for us, right? How do you do that? Right? I can afford a chair, but I need a Pixar thing. Not so easy. Right? Now, another challenge, and this is where you go crazy when you start thinking about these dome theaters. Right? You have to do fisheye projection, and you have to do that in 180 degrees fisheye, 8K stereo, 60 frames per second, and if it's stereo per eye, that's pretty demanding to work on. And that entails a way of thinking about your visual representations in a way that we have been looking at for more than 25, 30 years, trying to figure out how to do this. And in particular, if you want to do stereo, you know, in a fisheye environment, in a 360-degree system, left and right is not really well defined. So you have to think about where is left, where is right. And our solution to that is actually basically render 8 different camera views, 4 for each eye. And then we have a stitching algorithm that puts some sort of reasonable stereoscopic viewing back together again. The dome theater that we will experience tomorrow is unfortunately not in stereo, but if you see it in stereo, it's actually massive stereo that helps you to resolve some of the depth issues, some of the complex features that you might want to really convey to the audience. Now, you have a couple of choices when you do the rendering. And you will see examples of these in just a little bit here. And the examples, the way that you can do this is, of course, you can use standard tools. You can do offline rendering. You can use V-Ray or any of those other renderers that are out there. But if you're looking at the 8K format in stereo, it can take several hours just to render one frame on our cluster with 16 nodes and 512 cores. So you might want to consider going real time, which we do in some of our projects, and real time is really exciting, but it's not really predictable, because real time is not so secure. It depends on who's driving the application for you, and all of those things, so the visual quality may not always be there. An alternative is, of course, going hybrid. And this is really changing the way that we are working, and you will see examples of this, how hybrid rendering, when we're actually using a real-time application or even a gaming engine to drive the whole development process to generate some really nice content for dome theaters in a much, much shorter time, orders of magnitude shorter than if you do a pre-rendered material. Another challenge that you have to deal with when you start developing content for planetariums or for dome theaters, if you think about traditional way of making videos and movies, when you have a nice frame, forget it. Because you have this vast surface, the real estate of your screen is enormous, and you have no frames. So the traditional shots, the sympathy shots, the cuts, everything that you're used to doing in traditional filmmaking, forget it. It doesn't work. There are so many different things that you need to think about when you only have a little square of attention in the middle and you have this huge ambient context around you that you need to think about and fill with interesting material as well. But not intrusive material, because it's material that's just there to enhance your experience because you're focused on that square in the middle there. So it's the camera motions, the cuts, the localization, the pace of the story And you will see that in a little bit. It has to change a lot. So over these past 25 years that I've been working in this medium in a dome theater, we've been developing a couple of design principles, and I just want to share them with you. They're not published anywhere yet, but it's probably something that we need to do eventually. Now, one of the first things you have to think about is to only use that fraction of the dome. Use that fraction of the dome for the content that you want to show to make sure that the audience is following you. But use the ambient context for some interesting stuff, surprises, that adrenaline kick that you need to give to the audience as well. So use it intelligently, but it's also mentally very hard to translate when you're doing the design and the storyboarding in a fisheye projection to think about where is it actually happening in the dome. If you don't have access to a dome theater, it's actually a pretty big challenge to understand, to mentally transform a fisheye image to an immersive experience. Now, another thing that you will realize as well is that if you were to show these things on a flat screen, it's painstakingly slow because we slow down everything by a factor of 3, or maybe even 5 in some cases. Because the visual speed of everything when you're surrounded by a dome surface, the visual speed is tremendous. It goes really, really fast. Now, another thing is the camera moves. We work a lot with camera moves. The camera keeps moving all the time, continuously moving. If it stops moving, you will see the curvature of your dome. But if it keeps moving, your illusion is there, and you will perceive everything as being in 3D, even without the stereo. Another thing that we have to think about is how we move the camera. Fast zooming is fine, you can do that, and you will see that in a bit. Pitching is maybe fine, rolling never. If you roll the camera, you're at the risk of having an audience that's not feeling so well. So don't do that. Color contrast is an issue. In a dome theater, you have a lot of scattering of light, and it washes out the colors. So be very, very careful in the color contrast, the color scales that you're using when you produce content for domes. And use the sound for context and cues. Extremely important to localize that things are happening in time inside of the dome. So these are some of the principles that we have worked on. Now, if you have those principles in place and you want to start out by producing content, of course you have to have a production pipeline. And that production pipeline always starts with the research component. And in our particular case, what we always do is to look at what is the research out there, what is the state of the art in research, not only in the application domain that we're looking at, but also from the visualization perspective. And you know, back in 2017, when we started out this project, what I did was to see, if we're going to make a story for the dome about the chemistry of life, what kind of stuff is out there in terms of the visualization community? And you know, I found a couple of things. Sorry, here. Now, there's tons of research out there, wonderful research on medical and biological visualization, molecular visualization. Here are just 5 papers that I think were sort of foundational and inspirational for me when I was trying to figure out how are we going to go about and make this story about the chemistry of life. And it goes back to 2012, when there are some papers by Christian Hege, well known in our community, and his team. The simple realization that when you render molecular material, it keeps repeating itself. So you can instantiate on the GPU, you can instantiate the same molecules over and over and over again. And this was refined in other papers coming out in 2013 by Martin Falk, who's working with me, Michael Cronin, and Tom Ertel, they're all here. And the Megamol system that came out in 2015. At the same time, in 2015, CellView came out. And CellView was really a critical component in the whole discourse and the whole idea behind the chemistry of life. And Drew will tell you more about this and how the convergence of CellView and his ideas turned out to be one of the keys to success. But more recently, also, stuff like embedding the storytelling inside of the molecular visualization has been really inspirational. So these are just a few snapshots of how the visualization community is contributing to this work that we're doing to communicate science to the general public and do it in an exciting way. And of course, The other thing that you realize, if you want to make something about the chemistry of life, you have to bring in Drew Berry. And I'm so happy to have Drew here today. His animations are just amazing. So go out to his WEHI homepage and look at his animations, and you will be flabbergasted by some of the things that he's been doing over so many years now. So putting the team together in 2017, this is what I did. So I contacted Drew, I contacted Ivan and the CellView team, connected them with researchers on visual learning and the production team at the Visualization Center, and all of a sudden we had a project. The project is the chemistry of life. And if I'm going to give you the story of the chemistry of life in just a few seconds here, it's really a story about human life. It's a story about what's required from this planet to support human life in terms of things like the photosynthesis. But it's also a journey through the scales, because we have to position the human spatio-temporally in the right place to be able to perceive and appreciate these very complex things that we're going to look at. There's also a story about how can we do this? How can we humans actually understand these things? What kind of equipment do we need? What kind of advances in science have made this possible for us to actually understand things that are so small down at the nanoscale? And then bring you down to the mitochondrion, where much of the work that you're going to see is happening, and the adenosine triphosphate, the fantastic molecule. And then trying to convey the notion that this happens billions and billions of times inside of your bodies as we speak. Now, I just wanted to show you a few sequences that illustrates this idea. Now, we start out in the movie with a park, and it sort of creates a nice feeling of being inside of a friendly environment. And here we do the transition in the powers of 10 scale. And so we start out with things. I've recently learned that bumblebees are not okay in Australia. But we use bumblebee, we use an apple seed, and we're progressively going down in this sort of the powers of 10 journey down into the microcosmos. It's a salt crystal. It's the smallest thing that you can perceive with your sense of touch, is shown here. And we're going further down, and you see an egg cell, and eventually you're going to hit the human cell. What we're doing inside of the dome, because one of the things that always happens when you do these powers of 10 journeys is that you lose the context. Where did I come from? How big are the things behind me? So you will actually see in the dome that if you turn your head around, you can see all of the context all the way around you as you're spiraling down into the microcosmos. And eventually we reach the human cell. And this is sort of the contextualization of everything that we're going to show at the molecular level, because here we cut up the cell, and here you can actually see backwards. You can see all the other scales behind you, and you see the organelles inside of the human cell and the mitochondrion that's floating around in there. Now, one of the key components in the technology that makes it possible for us to actually understand this shorter nanoscale is, of course, the notion of light not being enough. And we're trying to show that by showing light waves. And here's a light wave, and here's a little protein sitting right in there in the molecule. And the light wave is so gigantically, and the wavelength is surely not enough to resolve that kind of detail. So instead of using light waves, we introduced the notion of electrons. And the electron wave is going to come here. And when the electron wave, with the shorter wavelength, hits the molecule, all of a sudden we can start sensing that. So in just one short little sequence, we're explaining why you need electron microscopes and why you need to freeze the proteins down to close to the absolute zero to actually bombard them with electrons. And this is the notion of the cryo-EM instrument. And the cryo-EM instrument, we have modeled, of course. And once again, one of those transitions that we can do in the immersive media is that we're turning into electrons ourselves. So we're going to fly in through the microscope and become electrons. There. The sound should have been a bit louder there. Okay. Now, when you're inside of the microscope, you realize that what happens down there is that those electrons are hitting the proteins, and it's hitting multiple instances of the same protein. So you get the same protein in different angles, different configurations, and you get thousands of images of the same protein. And if you use the medical visualization and computer tomography, you realize that then I can just put all of these back together again. And all of a sudden, I have a density volume of the protein. And that's all there is to cryo-EM, plus a little bit of more details. this is the perfect point for me while I'm showing this protein here to hand over to Drew, because now we're in the world of Drew Berry at the nanoscale. Drew, take it over.
Great, thanks, Anders. Thank you. It is such a delight and a privilege for me to be able to present to the visualization community. I really do appreciate the invitation. My name is Drew Berry. For the last 28 years, I've worked at the Walter and Eliza Hall Institute of Medical Research, also known as WEHI in Australia. It's just the other side of the CBD from here. It is Australia's flagship for basic research into infectious diseases like malaria, autoimmune diseases like diabetes, and many forms of cancer. And I leave this slide in here because this artwork, it's a 6-story artwork that we have installed in the facade, plays a non-stop roster of real scientific images. And the idea is for it to be a beacon to attract people's attention, but also to present data in a new kind of way, and show people what's going on inside medical research on the outside. I'm going to talk briefly about the pipeline I used for creating molecular visualizations and how we tackled the problem of creating content, molecular content for the dome. This is our pipeline. The biggest part and most important part of all of our animations is the first stage, is the research. We access the scientific literature, we do a literature review, we access all the different kinds of forms of data, the protein data bank or AlphaFold, molecular dynamics simulation, tomography, all forms of data. We bring that into our animation software, which in our case is Maya, which I've been using for over 20 years. And that's where we integrate all these forms of data, multi-scale data, different kinds of data, and determine our storytelling. And now we're currently exporting to Unity game engine for rendering and creating imagery. So all the animations play in real time in game engine. And that essentially is the punchline of our solution for creating giant content for the dome, was because of the rendering of imagery was going to be far too time intensive and expensive. But if we get into a game engine on a normal gaming PC, we can see these animations in real time. And then we create a virtual camera to record them. So we're outputting to movies or to augmented reality or virtual reality from these same sets of data. So the model or the science I'm going to talk about today is ATP synthase. It's one of the major 5 complexes of the electron transport chain, of which this figure is my favorite illustration from the scientific literature of the electron transport chain, where they have, so the blue box going across the middle there is the inner mitochondrial membrane, and they've placed all the different complexes, protein complexes of the electron transport chain from complex 1 on the left to ATP synthase on the right. And what they've also done is overlay the chemistry, the reactants coming in, the chemistry that's happening, and the products that are coming out. And it's my task to reconstruct what this chemistry would look like, but also the landscape, where this is actually occurring, and what it would look like if you could see it. All of our animations start with real scientific data, and a classic one is to use molecular dynamics simulation, such as this one, which is a ATP molecule surrounded by water molecules on the left-hand side, done as a molecular dynamics simulation. And then what we do is then extract out the ATP out of this simulation, and that is the basis that we'll then use to populate our animation. So all the small molecules have gone through this kind of a process, and then wherever possible, we'll incorporate real scientific data to make this stuff come alive and be as realistic as we can potentially make it, but to make it also engaging but meaningful for people. So it's a whole juggle of considerations. This one is actually of, it was for a different animation, but is of the lipid membrane. And what you have, the molecule in the middle there is coenzyme Q, quinone. And this was for a electron transport chain citric acid cycle animation. But this simulation of the lipid dynamics, we also incorporate directly into our animation. We sample these molecules, and then we'll populate with instancing and in-game engine from the GPU in real time. Our membranes, our proteins, our molecular dynamic simulations, that's all done from the GPU. but we sample from real data. This is a beautiful paper about the ATP synthase enzyme complex. It's from a bacteria system from a number of years ago, 2019. And what they were able to do is block or jam the ATP synthase at 3 different stages of rotation or activity. And they were able to then do cryo-EM, electron microscopy, to work out the 3D structure. So they had 3 different frozen moments of ATP synthase. And in this case, on the right-hand side there, they are blending between those 3 different states. We are able to access that data directly, and also there's actually six different steps of ATP synthase, and we can then make an informed speculation about what each of those other steps are like and build that into our animation. So this is my early stage ATP synthase, where it's my style of representation I like using. I personally prefer using the molecular kind of view, but I've also added in the reactants. So the ADP and phosphate are coming in. They're landing where the reaction at the active site in the enzyme, and then being pressed together to make ATP, and then fluttering away. So this visualization actually made for classroom use for high school teachers and for students to study. And it actually, so this is all built-in game engine, so it plays endlessly. You know, it's actually a 300 frame animation that plays endlessly, but still looks like it's a molecular dynamic simulation. So yes. Hopefully this will play. Yeah, one of the biggest challenges of representing Brownian motion or stochastic behavior is how long it takes. So this is of a kinase. It's a motor protein, a kinase that's, and the ATP is coming in and will eventually land in the active site. And the time steps here are nanoseconds or a billionth of a second. And even at this level, we have to wait around a long time for the ATP to get in exactly the right spot and for the chemistry to happen. And so this is obviously a really big problem for storytelling, because I need it to happen and so we can move on with the story. But if we sample at bigger scale, time scales, you would lose all this kind of data, this sort of the nature of how these molecules are interacting and moving around, the stochastic nature of that. So we compromise on that. We are aiming for the look and feel of this kind of a molecular dynamic simulation, but it has to be under animator control at all times so that I can specify exactly when events are going to happen. So see, ATP didn't even get in there in the end. So this is how we are, our trick for representing stochastic Brownian behavior of all the small reactants, where so in this case, we have, this is actually succinate and ADP and a phosphate, a hydrogen ion coming in, and they're landing at that letter N there. The reaction happens, and then they're going away. And what's the trick behind this is how do you make it look like they're just Brownian style wandering around, land at the right location, the chemistry happens, and then they wander back away at the same sort of look and feel. And the trick we're using to do that is this, again, this is in animation software. This is in Maya. What it looks like is that we create a path, like a random walk path, or essentially it's a fractal curve that we then use that as a motion path for our reactants. And in Maya, why we do that is that we can then change, because like a fractal, we change a seed value and you instantly get a completely new curve. So you can have infinite number of these paths, but also I can make as a choice as an artist which path do I want to use, that so it gives people enough time to see the reactants as they come in, and really, I will always choose one that they wander around a little bit before landing, so you can sight it and get a sense of it. Otherwise, it's all a bit too crazy and messy, because you really do have to watch it with representing the molecular world. It can get overwhelmingly messy, and it just confuses people. You have to make it meaningful for humans and work within the framework of what they perceive. So that's just part of the challenge. But this trick has worked for 20 years, this sort of a pathway, and I have a number of other techniques that I use, but they're all far more complicated, and this one works really well. I can then just keyframe when I want events to happen. Next level of data for the ATP synthase, this is from an electron microscopy, where they're showing that ATP synthase forms pairs on the inner mitochondrial membrane and cause this kink, this bend in the membrane, which causes a negative charge. which increases the amount of hydrogen ions, increases the efficiency of this system. But all the measurements and angles and such things go straight into building the animation. Next level of data is this lovely paper where they're using electron microscopy showing that ATP synthase pairs form rows of pairs along the inner mitochondrial membrane. You have 5 different species there, and just looking at all the variations between species. So all that good stuff, I love it. It's so fascinating. Goes straight into animation. And so this is a version with the ATP synthase pairs. This was actually done as interactive on a website. So students could then press buttons and it would then reveal different parts, or a teacher could use this in the classroom. So here we have the ATP synthase, and we're now going to break it down and look at each of the individual components within the ATP synthase. So here's the active enzyme in the complex that actually does the mechanical movement of changing shape and pressing together the reactants. This is the inner rotational motor driven by hydrogen ions, and that's all put in there. This is just an early stage of building animations, but this is also designed to continuously play for high school teachers or university presenters, or for students to interactively play with. Next level of data, I'm just going to blast through this. This is from tomography, where they've reconstructed from slice by slice of electron microscopy images, and they're working out the 3D structure of the inner mitochondrial membranes and the location of the ATP synthase. And this is very important for setting up the landscape of where all this stuff happens, and certainly love this sort of thing. This is the most fun part of any of these sorts of productions, is actually reading the literature and just going, oh, that's awesome. So here's the animation aimed at high school kids in the narration, or the general public, to engage them. Yeah, so I'll press play.
All life on Earth depends on this tiny, energetic molecule, adenosine triphosphate, or ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA. Generating ATP for your living cells are mitochondria, electrochemical batteries that converts energy from the food you eat and oxygen from the air you breathe into ATP. So where does your ATP come from? Deep inside your mitochondria, rows of molecular motors generate ATP, the molecule essential to all life on Earth. Enzymes bring together reactants to form a chemical bond, converting mechanical energy into chemical energy. A ring of enzymes work in step, creating 3 molecules of ATP with each cycle. Inside the molecular motor, a rotating axle powers the sequence. The axle is attached to a rotary molecular motor, moved by the force of protons, pushing from the other side of the membrane. A difference in proton concentration propels the molecular mechanism. Synthesis of ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA.
So to explain the music and the choice of sound in this, I had a grant from a local philanthropy, the Telematics Trust, and so I worked with the high school that my kids go to, and their middle school choir, and my sound designer. We wanted to, because this is a pretty amazing system. Like, I find it awe-inspiring, and I wanted school kids to have a sense of wonder about this and get engaged, and I wanted to see if we could use music to actually give that sense of how incredible this stuff is. So that was kind of the notion of that. Yeah, but and this animation plays all in real time in Game Engine. That's the way it looks. It'll play endlessly. That the actual animation component, the entire thing, including the camera move, is all on a seamless loop. It'll continuously play. All the small molecules, everything. So that's really the power of this gaming technology. Yeah, and that same system is what you'll see in the dome. It's the same model, all the same detail. So then it came, 2011 was the first, I think I met Anders and begin thinking about domes and dome theaters. And this project has been by far the hardest, most biggest technical challenge, and also artistic challenge, because everything is so different in the dome in giant scale to even what you see in virtual reality. So it's been such a privilege to learn from the whole team in Sweden. I've learned so much over the years, and it's been and I'm so hopeful, and I expect great things for the Chemistry of Life film. This was an early test. This is from about 2014, I think, where just the question was, and this is the time the dome, I think, was 6K resolution. How do we render that many atoms on the screen? Was it possible to do it? And I had to find a pipeline, a software rendering pipeline to do that. And what you're looking here is blood plasma, just the big protein molecules. You've got some fibrinogen with sort of the nice long ones on the purpley ones on the side there. They're what causes blood clots. And you've also got a rhinovirus, a common cold virus. This is a very sick person if they got rhinovirus in their bloodstream, but it does happen. And there's some antibodies up in the top right-hand side there. So this was a simulation. I had to go to a render farm, and eventually this was actually rendered on a big supercomputer somewhere in the US, and what was going to take, the render was going to take about 10 days on my local PCs to render. It took 9 minutes on the supercomputer, which is fantastic. I had to wait for it to get in there, but it was 9 minutes, and bang, it was done. And then I had to download the files from their server back to us, and that was going to take 12 hours because of the size of the files. So it really wasn't optimal. And also, when you render, you wait all this time, you download it, and there's issues, there's problems. And you go, oh, damn it, and do it again. So, and it was going to cost hundreds of thousands of dollars to do this kind of rendering, and it was all disconnected and very frustrating. So we had to find a solution, and this wasn't it. Even though it was possible, we could do this with all the shadow and rendering that I wanted to do. This is I had a dome set up locally in the office. It was a 3-meter vertical dome. And it was useful initially for me to get a sense of scale, because I couldn't keep going over to Sweden to see it all in the giant scale in the dome. And I did go occasionally to tests here in Melbourne at ScienceWorks, but really you need to see it at scale to understand why things have to move slowly, because there's so much space for it to move around. Things are very different on a giant scale. So it was useful, but I sort of, I then switched to virtual reality to test most of my content. Then Anders put me together with Ivan Viola and his labs doing this incredible work. This is all in Game Engine, playing with all level of detail, like a David Goodsell style illustration of all these molecules. And I mean, this is from 2015. I saw this in 2017, 2018, and this is like just dropping our pipeline the last 20 years. This is our new way of going. It was fantastic. So we were initially, we adopted this system. He released it open source on Unity, or connected with Unity. We have now since developed our own pipeline, but it is totally inspired and influenced by what Ivan's team did. And this was the solution. This is at ScienceWorks Dome, and it's hard to get a sense of how big it is, but this is an early test from about early 2019, of the ATP synthase, that same animation, playing on the dome and rendered through Ivan's, the real-time Unity game engine system. And so it was proof of concept. It was going to work, but little did I know that COVID was coming. Lots of data, and I just wanted to flag one bit of data. There's a lot of stuff that we don't mention in the film. It's a very science-heavy film, and a lot of technology and how we do microscopy and how protein structures are worked out is there. But we did do a lot of other science that isn't specifically mentioned, and one of them is, and I care very much about, is my representation of photons. So I did 2 sequences. The vast majority of the film was done by Team Sweden. I had 2 sequences, these molecular and cellular visualizations, one of photosynthesis and the other one of ATP synthase. But of photosynthesis, my background is a botanist, as a cell biologist, so I care very much about photosynthesis, and I wanted to represent it correctly. And for photons, I used this paper as a reference for determining the size of my photons, individual photons. And so essentially the photon is half, so it's based on its wave function, and it's half of its wavelength is how long a photon is in its vector, based on its probability, and then its width is a square root of its wavelength. And so those measurements I used for creating my visualizations of photons. So here we are inside a chloroplast, and we're descending on the thylakoid membrane plate. So on the bottom there, you've got this tiling, which is the photosystems there capturing the photons. And this looks very different on the dome. This is sort of the dome footage, but wait till you see it there. There's ribosomes floating in the clouds there. In the background, you've also got plastoglobules and a starch granule, the big white glob there, and ATP synthase embedded in the thylakoid membranes. But the main point is we are mixing up time in huge, we have photons now, we've got ATP synthase is actually visible. We have to do that to make it all work in 60 frames a second so that people can watch it meaningfully. But that's the point of this. It wasn't just like a Star Wars reference of all these lightsabers going on. It actually, it was a purpose to why I represented photons that way. And I was going to play the trailer, or yes, for the film. So this is the trailer, the English trailer for the Chemistry of Life film. Hopefully most of you, or many of you, will be there tomorrow night. Yeah, go ahead. Actually, seeing that bit of footage reminds me, yeah, 8K resolution was tough. 60 frames second at 8K resolution was tough. 3D is actually the hardest part, because you have to worry about how far things are from your face, and particularly the molecular world. You can't have things fluttering in people's face. So the 3D, which you can either experience in virtual reality or go to Sweden to see it on their incredible domes. It was actually, that was the hardest part. You're missing out on that on the ScienceWorks dome, but really the impact of domes is their giant scale. That's really what makes the real impact. Yep, so there's that. If you want to see more of the WEHI TV animations that my team is producing, there's the QR code for our YouTube channel. Otherwise, you can go to wehi.tv, and you can request animations and download them from there. This is my team. Etsuko Uno, sitting over there. It's been with me 16 years. Etsuko works on content specific for the Institute. She's working on a proteasome at the moment, which is soon to be published. It is awesome. And then Justin Muir has been with us 6 years, and he is from the games industry, and he is the one who recreated our pipeline, and he's a magic man. He creates buttons. Etsuko and I are always so happy. We ask him a technical problem, and he creates a button, and it's all gone. It's all so good. So with that, I'll switch back to you.
Yeah. Can you get my computer, my presentation going again?
Yep.
All right.
Thank you.
I'm sure you understand that it's really been a privilege to work together with Drew for these years now, and trying to bring his content to the Dome Theatre. And I just wanted to wrap up a little bit about the movie. And it sort of ends with, you saw the ATP synthase that Drew showed you in his animations. And, it really is, it gives you this sense of wonder when you think about the number of molecules that we are generating, and to try to convey the notion of that. The human brain, this is sort of the end of the movie, the human brain is the vast consumer of ATP. Every single neuron in your brain is consuming 4.7 billion ATP molecules per second. And this is the notion that we want to leave the audience with towards the end, is that, wow, my brain is really powered by this chemical motor, the molecular motor that Drew is showing you. And it's happening billions and billions of times inside of your body continuously, and makes the neurons click and the synapses work and everything like that. And that's the feeling that we want to leave the audience with when you leave. I had the privilege to show the Chemistry of Life at the Immersa Festival in Montreal. And here's just a little video of that, just to give you the feeling for the size of the screen and how you can swim around in the molecules and feel really immersed. And in the Dome in Montreal, they have sacks that you can lie down on the floor and just be amazed by Drew's animations in the big Dome theater. So that was a cool event that we did over there. But the Wisdom project is not only about the chemistry of life. We're in the process of producing content for these domes that I showed you. And Chemistry of Life is the third production. The first one was called Making Magic, and it's all about visual effects, computer graphics, and computer games and how they work. And it's trying to disguise the mathematics and the programming to show to the kids, hey, if you want to make a computer game, you actually know a little bit of math and a little bit of programming is required for that. The only requirement they had at the center when we were producing Making Magic, and I told the people down at the reception, I'm going to make a story about visual effects and computer graphics, that there has to be a dinosaur. That was the only requirement, okay? Because the kids, they love the dinosaurs. So the story is all about how do you make a dinosaur? And showing all of those tessellations that you need to do, the rendering, the ray tracing, all of that is shown in that production. And then we have one about space that is based on the Open Space project, and Alex Bock, who is here, is our mastermind of Open Space. It's an amazing story about the cosmic scales. And now the third one is the Chemistry of Life. What's in the pipeline right now is one production on artificial intelligence. It's a huge challenge. How do I teach the kids about machine learning and language models? And it's a fast-moving target. So if anyone has any ideas on how to make immersive visualization show machine learning, come talk to me in a coffee break, because I'm in the storyboarding phase of that project right now. And then the next one up is one about climate and environment. So this is sort of my hobby project when I'm not sitting there writing papers. I'm really working a lot with these productions, and I think it's such a great thing to be able to do that. What Drew and I what we hopefully have conveyed to you guys is this notion that if you engage in science communication, in public outreach, it's really a possibility to bring science, state of the art, out into the general public to the benefit of the next generation. But it's also an opportunity to conduct some really cool research. For us in the visualization community, the challenging aspects that we have faced with the rendering, the connections that we made between the basic research in our community and the end users that are producing amazing results with the help of the stuff that's come out of the papers that we have published over so many years. And making those connections is so rewarding to do that. And it's really paving the path for the next generation of science centers and museums. And we're being contacted by so many different places that want to screen our productions, use our tools, and the Chemistry of Life is being sold and licensed all over the world at this point. So this is pretty cool. But primarily, what I wanted to leave you with is, this is really slow, that it is extremely rewarding and fun. And there is really an opportunity for the visualization community. So it's a little bit of a call to action. I know that there are many people out there working on visual learning, education, but bringing that to the Science Center and working with the museums can be pretty cool. And as one of the final slides, there's probably a planetarium near you. There's one here in Melbourne. There's about 100 million visitors per year going to these planetariums. 100 million. And most of the shows are astronomy. And it's time to bring other stuff to the planetarium, because it's an amazing immersive environment, and there are so many opportunities to work together. These planetariums, they're so keen on having contacts with researchers. Just show up there and say, hey, I'm a visualization researcher. I want to produce some cool stuff. I want to show my things in your dome theater, and they will be very happy to help you. So think about that. Reaching 100 million people a year could be pretty cool. Now, you have the chance to watch the Chemistry of Life, and of course we don't want to interfere too much with the banquet, because the VIS conference is all about mingling, interaction, talking. But if you do have a chance, give us 30 minutes of your life, and I hope that you will not be disappointed by spending that time on watching the Chemistry of Life tomorrow evening. So the only thing that remains for me is also, just like Drew, to put up a slide and the acknowledgement for the amazing group of people that have been working on the Wisdom productions, and in particular the Chemistry of Life. Here are a few of the names and the organizations that have been supporting us, and the Knut and Alice Wallenberg Foundation that had the foresight to fund us with a significant amount of funding to build spectacular dome theaters and fill them with nice content. And thank you very, very much for listening to Drew and me today. Thank you. Good. Do we have time for questions? Or?
We have a few minutes for questions. Thank you so much, Drew and Anders. If you have questions, please come up to, there's microphones in the middle of the room here. Please come up. Perhaps while people make their way to the microphones, I'll just explain what's going on down here. So this is Natalie Overdiest, who is a PhD candidate at Monash University and also a very talented artist who has volunteered to make beautiful visual notes of the keynote, capstone, and also best papers talks this afternoon. And we'll publish those online later. If any questions, don't be shy. Maybe I'll just ask one quick question. So, Drew and Anders, obviously the primary audience for all of your animations and visualization work is the general public, and communicating to a vast audience about these really complex topics. But I'm wondering if the visual techniques, the animations, and the visual languages and things that you're developing, I wonder if they're feeding back into the scientific community in any way.
I didn't hear the question.
Oh, yeah, sorry.
I'm wondering whether the techniques that you are developing to communicate to such a wide public audience are also feeding back to the scientific community in the way that they prepare their own presentations and visualizations.
So I'll have a go at answering that. The answer is probably not really, because really the issue that my work addresses is the huge tsunami of all the data and discoveries that are going on right now, all the data as you guys are all wrangling, how necessary it is for the public to actually understand and appreciate what they're what is being discovered, and particularly in medical research, such as cancer discoveries or whatever it might be, to have the public come along on that journey and understand what is being discovered, what a treatment might be, what our new understanding about this or that mechanism is like. So that's really the role of what I'm doing. And behind the scenes, a lot of the tricks we're using are tricks from cinema and special effects. It's there to make it look like it's something it's not. So the movement example, for example, the Brownian motion I was describing, I don't think that would be appropriate, or maybe it would be. A random walk like that could be appropriate for some research, but it's not really the purpose of what we're building the tools for. And I do strive, my team strives to reflect exactly what's in the literature, what's being described, usually just in the text and figures, and we translate that and transform it into a visual narrative that the audience can watch. So it's an artistic endeavor, is what we're doing. Yeah.
I'm actually seeing the arrow going a little bit in both directions. There are examples of researchers that come to a planetarium, and we've done experiments with astronomers to show large amounts of data, galaxy data, for instance, and for the first time being able to grasp the size and do that kind of experimentation. So we're actually seeing a cross-fertilization of the tools that we are developing for science communication back into the research practice in those communities. But I think fundamentally it's also the challenges that we face as visualization researchers in terms of making those tools. And also, I mean, just imagine being able to instrument your installations in a museum with a few million visitors per year, the kind of data that you can extract out of that and the kind of research that enables for us. So we see it as a research laboratory in sort of a DevOps situation with visualization research and public dissemination. Right.
Thank you. And we do have a couple of people who have made their way to the microphone. I can see 3 people queued up there. We've probably got time for just 3 questions. So please introduce yourself and ask your question.
Yes, great talk. My name is Francesca Samsel. I'm at the Texas Advanced Computing Center at UT Austin. And we work with a team also at the University of Minnesota. And together, we're working on planetarium exhibits, and we've got it running in Unity and, you know, with ParaView and all the big data and all that kind of stuff. And we can fly through it. So we can do all that. But I'm struggling to find any guidance on and we have the fisheye projection and that sort of thing. But I'm getting, so this is a technical question, I'm sorry. I'm getting all my images around the rim. And I'm wondering if there's any source material you could recommend that would help me to be able to figure out how to compose those images so that my data doesn't end up, you know, just on the rim. So I'm looking for source material.
I can't hear what she's saying.
Yeah. Quite close to the microphone. It's pretty hard to hear from up here.
Oh, can you hear me now?
That's better. Thank you.
Okay. I'm looking for resources. Okay, so we have it up in the dome. We have it in Unity. We have the data. We can fly through the data, but and we have it in fisheye projection. But I can't control that all of the density of the data is around the edge of the planetarium. So I'm wondering if there are any source materials of people who build things for planetariums where I might go to figure out what I'm doing wrong.
If I get the question correct, it's that the data lives on the perimetry and that you're not using that much of the I think that's an artifact what you're perceiving when you look at the fisheye projection to some extent, and we're struggling with that because, you know, the pixels, they're varying in size across the dome surface, and as I showed you, the sort of the little square frame that you have is where you need to focus on the context and in particular if you use stereoscopic viewing in a dome theater, you're limiting the field of view quite a bit, so you still have to stay pretty focused on that thing, but as I said, it's sort of when you actually get into the dome and you're seeing the data surrounding you, there is an additional cue in terms of size, in terms of complexity, in terms of resolving things, and that's not necessarily living on the edge. It's happening right up in the middle, and you have, you know, the famous polar pinch of things going up in the middle of the dome. So I don't know if I'm addressing the question.
Yeah, no, that is. But I might email you.
Okay, all right.
Thank you.
Michael Krone, University of Tübingen. Thanks for that very inspiring talk. And obviously the dome projection is a very good medium for this kind of content. But have you thought about, or have you considered, since you have all this content and this technological pipeline now in place, have you considered also using that for VR experiences? I guess technically the transfer would be relatively straightforward. So how do you think? What do you think about having this as a sort of say personal immersive experience?
Michael, why don't you come down here and just post the question down here, and I'll read out. We can hear you.
Hearing the echo.
So just come down.
Yes, we have that movie in VR, and what's interesting is that you do capture all the 3D effects, which is quite its own impact, but it feels very different to the dome. It's really quite startling. So it works very well on. I have the Quest 3 here, and it works great on the Quest 3 as a movie, as a 3D movie, and you're immersed in the world, but it's very, very different feeling to the giant scale. Yeah, it's two different two different experiences.
Was that the question? Okay.
I think
Okay.
so. We can hardly hear like the. No, it's not you.
You might need to really speak up and project into the mic as much.
All right, great. Can you hear me? No?
Yeah.
Wow.
Speak slowly and clearly.
Okay, I think we're all getting blasted back here. All right, my question is the following. First of all, amazing work. You're so far ahead with this work. There are many others here who probably want to catch up and do similar things. And so for me, the big question is, how do you measure the impact of this kind of work? What is sort of the outcome that you try to capture? Is it number of people who've seen this, or can you go beyond that?
It's a very good question. And how do you measure the outcome of a science communication effort? How do you affect the audience? And at this point, we have those surveys that we conduct with, the learning outcomes. We have classes coming to the Dome. We try to assess them before and after the experience, and that's formed some sort of baseline for an understanding of the impact that we're having. But it's primarily, for me, it's more anecdotal evidence. And I'm just going to share with you an anecdote. Just a few months ago, I got a message in my LinkedIn inbox, and it was from a girl, and she said, I just started doing my PhD in cosmology, and it's all because of the presentation that you had in 2015. And I think that's the reward that you get when you do that science communication. And the more sort of evidence that I get in terms of that kind of experience, people that have changed their mindsets, have realized something new, and they start thinking about data, thinking about science in a different way after having have one of those science communication experiences. And hopefully, Chemistry of Life is going to do that to the next generation of people. There will be lots of Drew Berrys 20 years from now, hopefully. But that's the thing that we really want to try to measure. And if anyone has any ideas and insights on how we should measure learning outcomes from these experiences, I'll be really happy to talk about that. And as I said, we are going to have millions of data points pretty soon that we can collect from these experiences. But you are the judges of this after tomorrow night. When you had a couple of beers after the Dome experience, tell me what you think, for real.
Actually, I will say one thing. I
Right.
wanted to say. For those of you who have a more gentle constitution like mine, who on a small boat feels a bit nauseous, the start of the film is a bit of a roller coaster ride. It's very exciting, but some of us might, find it a little bit too much movement. If that's happening for you, just close your eyes and everything's fine. It works for me. So
I disagree.
It's just very exciting at the start. We're zooming around, so
I disagree completely, because as a filmmaker for Domes, you know that half of the audience is going to feel pretty tired because they've had a long day and they come into this dark room, and some of them are going to fall asleep if you don't wake them up. So that sort of roller coaster ride at the beginning is the adrenaline kick that you need to stay awake for the rest of the show. So, all right, okay.
Okay, please join me in thanking Anders and Drew one more time. Thanks. Thank you. And I think that they're both staying around for the afternoon sessions and the show screening tomorrow night. So please approach them with your questions then. We will start the afternoon sessions on time at 2:00. Sorry for going into your lunchtime a little bit, but I think it was very worth it for this wonderful talk. See you soon. Thank you so much.